Thank you. I've done a lot of things. I hope I can forward the slides. That's the challenge. So this is a little bit of a different session. It ties in well tomorrow into some of the theranostics, and I appreciate the opportunity to speak today, Friday afternoon. And this is a nice, I'm also the session chair, and just saying I appreciate the involvement. There's two scientists from the national laboratories, one from CNL, the other from Triumph in Vancouver, and we also have a representative from a Canadian industry related to alpha. So thank you to the other speakers, and thank you to the audience for your interest. So I'll talk about two things. These are rather short presentations, and this is actually combining two topics. First is the question of, what's the interest in alpha? What makes alpha so special? Right now we have beta therapy primarily. I-131 would be the prototype of that. And we do have one alpha for the radium-223 therapies, but what is the buzz about alpha? And the second thing will be a kind of a review. We did a systematic review of papers that have been published about alpha therapy, just to give everyone here a little bit of a taste of what's being looked at, which radiopharmaceuticals, which radionuclides, which countries are involved. So I think you may find that as an interesting introduction. So I have no financial conflicts here, and this is like an inverse of what we normally see, which is basically assume nothing here is Health Canada or FDA approved unless otherwise indicated. So as I mentioned, what is the emphasis on therapy with alpha emitters? Where is that coming from? Why should that be better than anything else we do? What are the potential strengths and weaknesses of our alpha emitters? Sorry. Okay, and we'll have to start with how do radionuclide emissions kill cells? So if the energy is absorbed in the cell, we have free radicals formed, and these end up leading to DNA breaks and ultimately to death of the cell. The advantage of alpha particles is they have a denser energy deposition, which leads to a greater lethality. And what's appreciated today is targeted cells also can affect their neighboring cells by a phenomena called the bystander effect. So in many times this is useful because your targeted therapy may not attack or hit every cell in the tumor, and through this bystander effect, you can spread your lethality to the neighboring cells and thereby kill a tumor. There's also a phenomena called the abscopal effect, where targeted cells can also, at a distance, affect other tumor cells. And we'll go over that briefly. It's a relatively new concept, something I had not learned about in medical school. It's a fairly new understanding. So if we look here at our targeted treatment, you can see this is a schematic of a cell. The two yellow cells are adjacent cells, and there's a little lightning bolt hitting the cell or hitting the membrane, and that refers to the absorbed energy. So it can either destroy the DNA directly, it can destroy organelles, it can interfere with the membrane, it can lead to oxidized radicals, which in turn do damage. But what this also shows is the bystander response, that either through ion channels, like in our upper right-hand side, certain toxic byproducts are affecting the adjacent cell, or it may be through soluble modalities, which are affecting the bystander cell, which is at the bottom of this cartoon. The abscopal effect is something new, as I indicated. It's seen more with higher amounts of radiation, so this is greater than 2 gray. And in this schematic, the woman has a breast tumor as well as distant disease. And by targeting the breast tumor, there are certain reactive entities that are circulated through the blood. Here they're called damage-associated molecular patterns, and these have a way of triggering the immune system. So it's a interesting concept that if you do have distal metastases, by irradiating the primary tumor, you can also treat some of the distal metastases. If we look at the therapeutic radionuclides we know of today, they pretty much define this straight line. And on the x-axis are the energy of the of the emissions. These are all beta emitters. On the y-axis is the range in tissue. So as you move from left to right, we're dealing with more energetic emissions, and they also perforce have a larger range. So I mentioned a minute ago, it's good to have a bystander effect so that you don't have to target every cell. On the other hand, you don't want a large range. Here, the dotted line shows a typical cell diameter of about 100 microns, and you can see all these emissions have a it's a log axis, they have a much larger range. So you're not only destroying the cell and its neighbor, you're destroying a whole region of tissue with each of these potential emissions. Tin, which is on the bottom, is a conversion electron, and you can see why people favor it because of all of them. It has the smallest radius. So these are all for different types of beta emission. On the other hand, if you look at alpha, it's really a different curve altogether. The alphas are where that little red dot popped up. And so for this, so typically they have energies between, let's say, 2 and 10 MeV, and the radius, instead of being 1 or to 10 millimeters, it's on the order of about 0.1 millimeter. And here I just plotted for the main emissions for 7 different alpha emitters. And you can see they're all clustered together. And when we look at it on our original graph, they're much below the beta emission. So as I said, this is really kind of a game changer, that you're able to impart a lot of energy in a much smaller radius. So the effect to normal tissue is much less, and we'll see that in a few minutes. So to summarize, beta particles have a low linear energy transfer on the order of 0.2 keV per micron, and they have a relatively long path length of between a third and 12 millimeters, which is several hundred cell diameters. So they're killing, or they have the potential of killing, many more cells than the ones that are targeted. In contrast, alpha emitters have 8,000 times the mass, 2 times the charge, and they have a very high LET. It's 80 to 100 keV per micron, so that's 500 times as much as what we saw with the betas. You end up depositing 48 MeV within a small radius, which is a few cell diameters. And they therefore have the potential that a single particle can kill one cell. The relative biological effectiveness is 20 times that of a beta particle. It doesn't depend on oxygen concentration, so these other phenomena are not important, and it has a reduced bystander effects. So that, that kind of answered my first question. Why is alpha so good? It's really a very targeted process with a very high deposition of energy. So what are the radionuclides we can use for alpha therapy? There's about 2,500 isotopes in the chart of nuclei, and only a few dozen are suitable for medical use. And we look at things like the half-life and the mode of decay. These are important to us, whether it's alpha, beta, or gamma decay. And, are there any chemists in the audience? I think there's at least one. Chemical properties are very important. If you even look at technetium, much of the advances in nuclear medicine have to do with chelating and binding technetium to take those radionuclides and make them useful in a chemical form. Finally, we will touch upon, and some of our speakers will touch upon accessibility and availability. It's not obvious that these things are available, and we'll look at that in a little bit. So this is the chart of all the radionuclides. Of course, I can't see it well, but so the number of neutrons is on the x-axis, the number of protons on the y-axis. All the radionuclides known to man, except for one or two, which may have been derived since this was printed, are the yellow and turquoise and purple boxes. The turquoise boxes are the stable isotopes, and you can see there's relatively very few of them. And depending on where you are on this curve will determine the type of decay that goes on. So if you're in the lower right-hand corner, relatively speaking to the line of stability, you have too many neutrons. So how do you become stable? And when you decay, what is the mode of decay? Sorry. So there you have emission of a beta particle. So if a neutron kicks out a beta, it becomes positively charged. So you move to the left and you move up one, and you therefore move towards the line of decay. If you're to the left of the curve, you have too few neutrons, and the way to do that is either to capture an electron or to give off a positron. And what we're talking about today are the more massive nuclides, and they're in the top of the image. And what they do is they give off an alpha particle. And this just shows you by schematic, if you look at U-238, how it would decay. So it gives off the helium nucleus, which is a beta particle, and it decays down to thorium. In the meanwhile, that's excited, and it'll give off a few other gamma rays and so on. And oops, sorry, I was too busy looking at the numbers. So you can see it actually in my little chart, the upper right-hand corner. As it decays, it goes down 2 and 2 to the left. Eventually, when it gets under the horizontal line, it too becomes stable. So that is our alpha decay. How do we get the radionuclides we need? This is a very interesting, I love the title of this article, A Nuclear Chocolate Box, the Periodic Table of Nuclear Medicine. And, you know, this is a scene from Forrest Gump, if you remember the chocolate box scene. But so the idea here is we have when you order chocolates, you can't get something that isn't in the box that you bought, right? So we have a limited box of radionuclides. How can we pick the best for us? You know, we do have a limited number of possibilities. Okay, so if we look at our chart of elements, these blue dots are all the beta emitters, and some of our friends are here: lutetium, I-131, samarium, strontium, yttrium. And I've overlaid here the alpha emitters that we have, and our good friend radium is to the left. But some of these newer agents that we're going to talk about today, actinium, lead, with the right radionuclide, are on the chart as well. So these are all the elements that have a radionuclide of interest in therapy. Finally, we have the conversion electron here indicated in green. So this is a list that Dr. Skoris put together a few years ago of all the commercial ones that are either released to the public or still in development. And you can see the atomic number of 82 differentiates those that are beta emitters from alpha emitters. And we have a number of beta emitters that are marketed. These are the marketed ones. There's a little blue asterisk next to the drug itself. And there, as of today, still true, there's only one alpha emitter, which is our Xofigo, which is radium-223. That is the exception. That is the one that is Health Canada approved. Well, some of the betas are Health Canada approved as well. So if we just use that as an example, looking at the bone therapies, many of these radionuclides or radiopharmaceuticals you've had experience with in the past. Some are still marketed, some are no longer marketed. They're approved and or in limited use, and radium is the only alpha emitter which is come along. And if you look at the difference here, most of these betas have mean ranges on the order of several millimeters, while radium-223, the path length is less than 1/10 of a millimeter. Again, that allows us to target the cells we want and not widely affect the marrow or other normal cells. So looking at radium again as an example of an alpha, the half-life's about 11 days. Most of the energy comes out as an alpha particle. There's a little bit of a beta, a little bit of a gamma, and that actually is advantageous. It makes it easier to survey and to measure and clean up afterwards. There's a standard dose we give over 4 weeks, 6 times, and it's quite effective. There is a side effect is a certain degree of myelosuppression, which tends to bottom out at about 4 weeks and recover by 7 weeks, and it's primarily excreted in the gut. Radium is produced from an actinium-227 base generator system, and as I mentioned, there's betas and gammas, and here is the case scheme. So I just want to point out a couple of things. So we're starting on the upper left with radium-223. This is a test, chemistry test for me. Goes to radium, polonium, lead, bismuth, thallium, polonium, lead. But what's interesting and is different than what we're used to in the gamma world is some of these intermediate products hang around for a while. So it makes it a challenge in the biology. So even if you target the area you want, when it decays, a new chemical form, new element derives, which usually is not or may not and gives off energy. It's not constrained by the original chelator or targeting method, and it's free to wander around the body. So if you have some of these longer lived daughters, they can go elsewhere and do other things, and you have to take that into account when you do your dosimetry. So it's a different kind of a way of thinking about these products. You have to worry about their progeny as well as the primary targeting as well. And this is just a list of the gammas that are emitted. This highlights this idea of the targeted therapy. If you look at the little red cluster of cells, this indicates there's targeted radium-223 at the one yellow dot. If we're talking about the beta emissions, it's the whole blue circle, which encompasses the tumor as well as a lot of marrow. If we only are focusing on the alphas, and that's why we give the alphas, it's a much more constrained radius of therapy. So that's the idea, hit them hard and hit them in a very concentrated manner so you're not affecting normal neighboring cells. So we had the TAD11 conference here in Ottawa, and that was sort of my major introduction or involvement with the alpha therapy community, and that's here on Parliament Hill. That's the other part of Ottawa, which we're not talking about here today. And we had a beautiful conference. We brought together scientists from all over the world, and that was also published as a proceedings in the same journal that we're publishing this morning's conference in JMIRS, which happens to be a Canadian technologist journal. So this was the 11th iteration of this meeting. It tends to be every 2 years. It was held in Ottawa 2019. It was co-sponsored by the two national labs, Triumph and CNL, in combination with a bunch of international and national groups. And the University of Ottawa had a hand in it as well. We had almost 500 registrants from 26 countries. This was fortunately pre-COVID, and there were many abstracts, talks, and so on. We had the first ever clinical day, which was something I was involved in, and we published the proceedings. Fortunately, I just came back from March. It took them 4 years because of COVID to have the follow-up, but TAT-12 was just held in South Africa, and I'll give you a few highlights from that meeting. So this is the second part of my talk, the background I hope you have. And now where are we? Where are we and where are we going? So we did a systematic review of anything published to do with human trials. So not abstracts, published articles to do with alpha emitters. We wanted to understand the gamut of effort invested in the field of targeted alpha therapy, specifically human studies. We looked at English literature, published literature only. The fellow with me is Mike Sethke, who organized the South African meeting. He's a nuclear medicine physician in South Africa and a world expert in alpha therapy, and this is the title of our review. So we did a search strategy in consultation with the librarian who does these things. We looked in both Medline and Embase. We did from the beginning of these databases. Of course, alpha therapy is not that old. So we started at time 0 and we went up to November 2022. We looked at these 6 radionuclides, which are the basic radionuclides used in therapy: lead-212, bismuth-212, 213, astenine-211, actinium-225, and thorium-227. And we looked at things that related to human use, human trials in the English language. We did not include radium-223 because that's accepted. There's many articles that would have, and we were interested more in these things that are being developed. Okay, so let's look at the results. We identified initially 645 articles, and they were reviewed in full and abstracted. 67 met the inclusion criteria, so that means since the beginning of time until about a year ago, there were only 67 articles in the medical literature. Primarily, authors were from only 5 countries. Sorry, I take that back. Authors from 5 countries contributed 52 papers, and the leaders were Germany, India, USA, South Africa, and Sweden. And another 10 authors another 10 countries contributed another 15 papers. So it's very central, and as you might expect in a young field. The journals, the leader was the European Journal of Nuclear Medicine, which I think is no surprise. They have been leaders in many new radiopharmaceuticals. JNM had 13, Clinical Nuclear Medicine 9, Cancer Biotherapy and Radiopharmaceuticals 4, and 20 other journals had an additional 23 articles. So this is of some interest, which radionuclides. So 46 of the papers dealt with actinium-225, 11 dealt with bismuth-213, 5 with lead, 3 with astenine, and 2 with thorium. Tumor types, which I think is also no surprise, the majority were prostate cancer, 33 of the articles. So this is really an extension of what we've done in the beta world. They've moved it into alpha. So prostate cancer is a leader, and it became a leader here. The NET family were 19 different papers, ovarian cancer 4, hematologic papers 4, and 4 other types of tumors. Now, this is really somewhat surprising. Of all these papers, 20 of them were case reports. Many of them were retrospective results. There were very few prospective studies and no randomized trials as of 1 year ago. Published work focused on outcome, toxicity, predictive indices, which patients will respond well, a little bit on imaging, some on radiation safety and other related topics. 27 of the publications had less than or equal to 3 patients. So we're really dealing with a very early field. This is one of the more famous papers that came out, a very successful paper, and again from Dr. Sethke from South Africa, and it's looking at actinium-225 labeled PSMA 617, in patients with advanced prostate cancer. And picture tells 1,000 words. So here we have the imaging to show you the results. So it's gallium-68, PSMA-11, imaging to show the results of the therapies. So between the white images are little arrows with a superscript of the radionuclide alpha therapy amounts. So 8 megabecquerels, 7.5. So they did a reduced schedule here. And you can see the multiple tumors throughout the body, basically soft tissue foci melted away over a course of time. Here's another patient with bone predominant disease and also an excellent result. But, you know, it's not always like that. Here's a patient that had progressive disease that was not able to be stabilized, and the patient eventually went off trial to chemotherapy and succumbed from his disease. Just to give you, again, a little potpourri of other articles that are out there. This is a 2022 article from the JNM from India using actinium-225 labeled dotatate in NET gastroenteropancreatic type tumors. This is a large study, 91 patients. I can't read what it says, but sorry about that. I should have brought my binoculars. Here's another paper also dealing with gastroenteropancreatic neuroendocrine. Was this the same one? Sorry. Yeah, sorry, same paper. And this is just showing you the tremendous results that they did obtain. Here's a case where they're targeting gliomas with a bismuth-labeled chelated moiety, and here the injection was intratumoral. So this was published in the European Journal from Switzerland, and substance P was injected with local injection into the glioma. So just again, the scope of what has been done, only 5 patients were in this published trial. And here is a follow-up on the same group, 2019, and to the 10-year follow-up. Here's an interesting imaging paper. So as I mentioned, the daughters have different behaviors than the targeted radionuclide, and people for dosimetry are very interested in tracking that. So this paper from 2020 from Sweden, and I'll just show you an example. 2 patients, but you have 2 different energy windows with the summation on the rightmost column, early and late time points. And I mean, the imaging is awful, but you can imagine it gives you a sense, looking at the different energy windows, you can get a handle on where some radionuclides are versus other radionuclides. So this is a technique that's being worked on now. This is using population type data. They were trying to estimate secondary cancers that may happen after alpha therapy. Again, it's a retrospective type look. This is a patient to do with lymphoma. So there's a few, as I summed up earlier, maybe 4 or 5 papers have to do with hematologic malignancies. And this is an earlier paper, well, from 2021, looking at thorium-labeled anti-CD22, first in human phase 1 study from Sweden again, and IV NHL 21 patients. So here is a 2007 paper looking at metastatic melanoma from Australia. A lot of melanoma work comes from there. Again, 22 patients, which is considered a lot. I'm almost winding up here. Here's an anti-CD33 in patients with leukemia. This is quite early, 1999, and using bismuth-labeled humanized antibody, and in 9 patients, and there's a more recent follow-up from 2022. So the title is interesting. They call it alpha particle nanogenerator, actinium-225. And the reason is, I'll show you, on the decay scheme, there's multiple alphas that are being produced. So in a way, each radionuclide results in several alpha emissions and kind of multiplies the degree of cell killing possible. We've looked at safety, looking at our technologists who administer this, how much radiation are they getting, a paper from India, 12 patients. So in conclusion, it's very early in the field. There's many features of interest in the TAT use, but a lot of weaknesses, very small studies, retrospective studies. And I think the involvement in industry today will make things a lot better. I'm going to just speak about this for 30 seconds. There is a bottleneck in supply, and we'll address this in the upcoming lectures. Oak Ridge National Lab in the US has been the main supplier of actinium-225, and they harvest about 800 millicuries annually, but the projected need is about 6 curies per year. So it's woefully short of what will be needed. And there are different methods of making alpha sources other than using these. Most of these are byproducts of reactors. So to sum up, alpha particle, tremendously potent therapeutic agent, 20 times more so than beta, very short path length. Proof of principle is our experience with radium-223. Uses may be limited by organ toxicity, lack of randomized controlled trials, and issues of availability. And there's been some early use in hematologic and oncologic application. Radiation safety is not excessive, and so on. So this is a tale of two cities, Ottawa, upper left, and Cape Town, South Africa, lower right, where the last two conferences were held. Thank you. We'll move on to the next speaker.